what? These specific kind of cells are having the capability to make different types of cells. Suppose I am having a particular type of cell, and if you know the different types of tissues are there, like we are having the heart cell, we are having the muscle cells, we are having the skin cells, we are having the bones, so different nerve cells, so different cells we are having in our body. So if we need to have a particular cell, then we can produce these particular cells from the stem cell only. You just see here, the figure has been made. You just see these are the stem cells. These stem cells will be collected from the source, and after that what we will do, we will put in this flask, and after that we will grow it, and in this culture flask what we will do, we will provide different conditions. In different conditions, these stem cells will make different cells. Suppose we need to have this heart cell, then we can differentiate these stem cells into heart cell. For suppose we need to produce this nerve cell, then we can produce the nerve cell from the same stem cell. Suppose we need to have blood cells, then we can produce blood cells from the stem cell. You just see, to by providing different conditions, the same stem cell is producing different cells, and these cells would be utilized in different diseases, such as paralysis, Alzheimer disease, cancer, and many more diseases. So you know that not each and every cell you can transplant from one person to another, and if the source of this cell is from the same person, then it would be so much beneficial to us. Suppose a person is going through a certain disease in which the person needs the blood, bone marrow. Then what we need to have, we need to have the stem cell. If this stem cell belongs to the same person, then it would be so much beneficial for that particular organism. Okay, so stem cell we are using for the therapeutic procedures. So we will see here, stem cells are those cells which have the ability to develop to many different cell types. So you have seen already that these stem cells are developing in different lineages by providing different conditions. Okay, these are obtained from embryo as well as adult human. So these stem cells, I told you, different sources we are collecting these stem cells. So sometimes these stem cells are collecting from the embryo, and sometimes we are collecting these stem cells from the adult human being. Okay, adult organism, we will say. Okay, so if we want to get this stem cell, then two sources are there. Either we can collect it from the embryo, either we can collect it from the adult person. Okay, they have been utilized to repair and replace the damaged cells during the stem cell therapy. So you must have heard about this particular procedure, stem cell therapy. So what we are doing, we are utilizing all these stem cells for the therapeutic use, and we are replacing the abnormal cells, we are replacing the damaged cells by all these stem cells. Suppose a person is having the blood cancer, it means the person is having the defected bone marrow cells. Then what we will do, we will kill the damaged cells of that particular person, and we will replace the damaged, or we can say the abnormal cells, which are going through the cancerous abnormalities. We will replace it with the stem cell, and after that what we will provide some conditions so that these stem cells will be converted into bone marrow. And if these stem cells will be converted into bone marrow, then these are normal healthy cells, and these will start producing the normal cells. If the person is paralyzed, then the nervous system has been affected, and you need to know that nerve cells are not repairing. So if the nerve cells are not repairing, then from where you will get the nerve cells? By stem cell only. So what you will do, you will take the stem cell and you will produce the new nerve cells from these cells. So these cells are so much essential in stem cell therapy, and especially we are using these stem cells in cancer, Alzheimer, and paralysis. And what we are doing, we are replacing the abnormal or damaged cells with all these cells, and by providing different conditions, we can make different cells. Suppose a
Sir,
person's liver cell
sir,
is damaged or abnormal, then what we will do? We will take these stem cells and we will replace the damaged cells or abnormal cell with this stem cell. Do you have any question?
yes, sir.
What is your question?
Sir, an adult body also has stem cells, and if it has, then what is the need of preserving stem cells at embryonic
Actually,
state?
this is very much good question. You just see, as I said, that we can collect these stem cells from the embryo as well as adult human. But you just see that here in this example, one, two, three, four, five types of cells are there. It means I have given five examples. Means here this stem cell is it can form five type of cells. So if we will take if the source would be the embryo, then it can be differentiated in many more type of cells. But if we are collecting these stem cells from the adult person, then it will be converted into few of the cells, even though most of the adult cells are converted into a single lineage only. Okay, so here there is a foundation. If we are, and I'll let you know what is the meaning of pluripotent and totipotent. Then only you can understand. Totipotent means I'll let you know further in this class, in this chapter. Just totipotent means the cell is having total potency. If we are collecting the cells from the embryo, or especially from the early stages of the embryo, then these will be called as totipotent. You know that the whole embryo is being converted into different organ. You know that ectoderm, mesoderm and endoderm, three germ layers are there. All these germ layers are making different body organs. So if we are collecting these stem cells from the embryo, then we can make different types of cells. But if we are collecting the stem cell from the adult human being, then we can differentiate in few of the cells, and mostly in a single kind of lineage. And here, you know that one company is there in south which collects the umbilical cord from the babies. So suppose they are preserving the umbilical cord in -80 or the liquid nitrogen. Suppose if a baby becomes 50-year-old adult person after 40 years, and the person needs the particular cell. Suppose person is having a certain kind of cancer, blood cancer, or some any other disease. If that person needs cell, then this would become the source. Means the umbilical cord which is collected by the company will from there. That is a good source of stem cell. So from that umbilical cord we will collect the stem cell and we will grow it. After growing, we will provide a certain condition through which we can convert the stem cell into a particular lineage. So nowadays, in developed countries, persons they used to store their umbilical cord. Okay, so we can preserve those. So those are the good sources of the stem cell, because if you'll become adult, then you will have very few type of stem cells in your body, and that will be converted into only few lineages only. That's why in case of paralysis or cancer, we don't get much kind of different lineages. So if you are taking the donor, if you are taking the cells from any of the donor, then somehow it will elect your immune response. So sometimes it will be harmful to that person, and there is a limitation. But if the stem cell is the source of your body only, then it would be so much beneficial. Okay, got your answer?
The stem cells do not trigger immune response.
Actually, most of the stem cells will not elect the immune response. And if this stem cell belongs to your body, then since it is belonging to your body, its genetic material is yours. It means it will never elect any immune response. But although if you are collecting a stem cell from another organism, another individual of the same species, then most of the time it will not elect any immune response. Okay, but if it is belonging to the same person, then it will be so much better. Now,
Okay, sir.
totipotent versus pluripotent. You will have to remember the term because based on this, most of the questions will be asked. You just see totipotent. So by the name, we can understand toti means total, potent means potency. So cells having ability to develop any kind of cell, even a fully developed organism like zygote. You know that the zygote is the single cell which is differentiated in the whole body. So after zygote to certain stages of the embryonic period, I told you early embryonic period, the cells will be totipotent. But after that, it will become pluripotent. Okay, so you just see pluripotent. If we are talking about the pluripotent, then pluripotents are the cells having the ability to develop many types of cells, not all. You just see for totipotent, for totipotent zygote, you know that it can be differentiated into the whole organism. The same zygote is making each and every organ, each and every tissue of your body. But when we are talking about this pluripotent, na, you will see the certain type of cells it will form. Suppose we are talking about the ectoderm. You know, the ectoderm will form the only certain organs only. If we are talking about the mesoderm, it will make certain organs only. Okay, so when it will become more and more, the cell will become more and more mature, then it will go towards the pluripotency. So totipotent, the example of totipotent is zygote, and the example of pluripotent cell is stem cell. So you will have to remember that when we are asking about this totipotent, then we are talking about this zygote only. If we are talking about this pluripotent, it means we are talking about stem cells. So stem cell will never be totipotent. Stem cell will be only pluripotent. You just see only few types of lineages it will form. So when we talk about these types of stem cell, so you just see four types of stem cells are there: embryonic stem cell, adult stem cell, induced pluripotent stem cell, and mesenchymal stem cell. So one by one we will see here. Embryonic stem cell. I told you the embryonic stem cell means we are collecting the cells from the embryo. So you just see this one is the blastula, and here you know this structure, and this one is the inner mass. So here what we are doing, we are collecting the cells from this inner mass. So if we are collecting the cell from this inner mass, then it will be called as embryonic stem cells, and these are having much more capability to be converted into different lineages. So here during the initial phase of the embryonic development, all the cells are totipotent, and stage of the embryonic development, blastocyst, a mass of cell arises in a hollow ball of cells called inner mass. You know the inner mass, and these cells contain pluripotent cells, they that make up developing fetus. So you know that the whole body of the fetus is formed by inner mass cells. It means these are having the stem cell which will be converted into different lineages, even though these inner cell mass have not been converted yet to the ectoderm, mesoderm, and endoderm too. So it is very much beneficial to collect the cell from here, inner mass. Okay, so these will be called as embryonic stem cells. So if anyone is asking the what is the source of embryonic stem cell, then your answer would be inner cell mass of the blastocyst. So from blastocyst we are collecting the cells which will be developed into different lineages, and here this source will be called as stem cell. Now embryonic stem cells can further be classified. You just see totipotent stem cell. Totipotent stem cells mean totipotent embryonic stem cells. It will be converted into different lineages, can be differentiated into all the possible types of stem cells. It means for each and every type of cell and tissue, you can make from this totipotent stem cell. So if you are collecting the cell, so if you will be so much lucky if you can get these kind of cells, totipotent stem cell. After that, after certain time point, what will happen, you will get pluripotent stem cell. So can be differentiated into any type of cell, means here not all the possible type of cell but most of the types of cells. Multipotent stem cell means differentiated into closely related cell types. Means closely related cell types means, suppose if we are talking about the muscle cells, so different types of muscle cells are there, cardiac muscles are there, skeletal muscle cells are there. So more or similar kind of cells it will form with multipotent stem cell. When we are talking about the oligopotent stem cell, so adult lymphoid or myeloid cells, can be differentiated into few different types of cells. Lymphoid means lymphatic system, or myeloid means RBC, WBC, and platelets. So blood and lymph cells, if the stem cell, if those stem cells which can produce blood cells as well as lymph cells will be called as oligopotent cells. You will have to remember these points. Unipotent stem cell, uni means single. I told you, if we are collecting the stem cell from the adult organism, then most of the time it will be converted into single lineage. You just see, can make or differentiate into only type of cells. It means suppose we are collecting the stem cell which is making only bone marrow cells. It means we can produce only blood cells only. So here, if the stem cells are producing in a single type of cell, then it will be called as unipotent stem cell. Now, the second major type of stem cell, I told you, is adult stem cell. So these stem cells obtained from the developed organism, or the human being, and from their organs and tissues. If we are collecting the cells, then these will be called as adult stem cells. These cells in body repair and replace the differentiated cells. So hemopoietic stem cells in the bone marrow, you know that the bone marrow also functions as a stem cell in our body, because all the time it is replacing the older cells with the new cells. So if we are talking about the bone marrow, so bone marrows are also considered as a kind of stem cell, because it is the mother of all the kinds of blood cells, so used in the bone marrow transplant. Okay, so suppose any of the defect is there in the bone marrow, then these adult stem cells or bone marrow transplantation is being done. Okay, now induced pluripotent stem cell. So by the name we can understand, induce means if we are inducing, if suppose a non-stem cell is there, if a cell is there which is not in the condition to be differentiated into another lineage, but if we are providing certain conditions, if we are providing certain nutrients, hormone, and some extra material, through which if you can induce the non-stem cell into stem cell, then it will be called as induced pluripotent stem cells. We are working on that, but yet we have to do a lot of experiment for that, because it is so much difficult process. If we can do this one, the differentiated cells or the cells, if we can convert into pluripotent cells, then most of our diseases will be cured by this. Okay, so induced pluripotent stem cell means the tissue specific cells have been converted into embryonic cells in laboratory. It is very much difficult. You just see, if we are taking the skin cell and if we are converting the skin cell into their mother cell, or the originator cell, or the progenitor cell, or the embryonic cell, then it is so much difficult. So the differentiated or the converted cells have the characteristics similar to the embryonic cell. So after this redifferentiation, what we will get? We will get the stem cell. Okay.